Biological hazards are a major type of hazards that are associated with food products and processes. Through this module, you will have a good understanding of the biological hazards in food processing facilities and be able to list some control strategies to prevent and control biological hazards in foods. There are three major hazard groups: biological, chemical, and physical hazards. Compared to chemical and physical hazards, biological hazards is the most reported hazard group associated with foodborne illnesses in the United States. Biological agents cause majority of the foodborne outbreaks in the United States. The most common biological hazards are foodborne pathogens. These pathogens can be bacteria, viruses, or parasites. The definition of pathogen is a microorganism of public health significance. Many microorganisms are not pathogenic to the general public, even though they can cause spoilage of the food products and economic loss. However, those microorganisms are not directly related to food safety, and they are not foodborne pathogens. Besides pathogens, prions, which are proteins, are the agents responsible for bovine spongiform encephalopathy, or BSE, also known as mad cow disease. Prions will not be covered in this module. It is worth noting that even some molds produce hazardous toxins called mycotoxins. Since mycotoxins are chemicals, they are usually chemical hazards but not biological hazards. Based on how foodborne pathogens cause illnesses, they can be put into two categories: foodborne infection and foodborne intoxication. For foodborne infection, a pathogen invades the body after contaminated food is consumed. In this case, the pathogen is in the food and someone eats the food. After that, the pathogen is in the person's body, invades the body. Some bacteria, such as Salmonella and Listeria monocytogenes, can cause foodborne infections. In addition to that, viruses and parasites only cause foodborne infection because they only grow in the bodies but do not grow in the food. Foodborne intoxication is usually associated with three bacteria: Staphylococcus aureus, Clostridium botulinum, and Bacillus cereus. Some Staphylococcus aureus strains produce enterotoxin. Clostridium botulinum produces botulinum toxin, which is a neurotoxin, and some Bacillus cereus strains produce cereulide, which is a emetic toxin and causes vomiting. Among all the pathogens, bacterial pathogens are a group of microorganisms commonly associated with foodborne illnesses and outbreaks, and many of them cause severe illnesses. There are two groups of bacteria, gram-positive and gram-negative, based on their cell wall structure. Examples of gram-positive foodborne pathogens include Listeria monocytogenes and Staphylococcus aureus. And examples of gram-negative foodborne pathogens include Campylobacter, Salmonella, and pathogenic Escherichia coli. Most of the bacterial cells are vegetative cells, but some gram-positive bacteria can produce endospores, which are a non-productive form of bacterial cells. Endospores from foodborne pathogens can be a safety concern because they are very resistant to some common treatments. Three common foodborne bacterial pathogens that can produce endospores are Bacillus cereus, Clostridium botulinum, and Clostridium perfringens. It is important to understand the factors that influence microbial growth because they will help us better understand the strategies we can use to control them. Some of the factors are intrinsic factors, which are the characteristics inherent to the food itself. Some important intrinsic factors include water activity, pH, and oxidation reduction potential. The oxygen availability is one main factor correlated with the oxidation reduction potential. Some foodborne bacterial pathogens, such as Salmonella, can grow with or without oxygen. Campylobacter needs small amount of oxygen to grow, while oxygen is toxic to vegetative cells of Clostridium botulinum. Extrinsic factors are factors external to the food. The most important extrinsic factor is temperature. Most foodborne bacterial pathogens grow more slowly at a lower temperature, while they are inhibited or even killed when the temperature is high enough. However, very high temperatures will be needed to kill endospores. There are three main aspects for controlling foodborne bacterial pathogens. One is to prevent contamination of food products. For example, good personal hygiene can help prevent contamination with fecal matters which could contain pathogens. Good personal hygiene can also help prevent the transmission of Staphylococcus aureus, which is a common organism that can reside on our skin and in our noses and can be transferred by touching. Effective sanitation practices, which should be included in the sanitation control, can help prevent the contamination of food products with pathogens from the environment, such as Listeria monocytogenes. Another aspect is to control the growth of pathogens in food products. This is important because some pathogens need to achieve certain doses to cause infections, and some pathogens need to grow in food products to produce toxin for intoxication. A common way to control the growth of those pathogens is to use low temperatures. Therefore, avoiding temperature abuse is very important. Most of the foodborne bacterial pathogens can grow from 40 degrees Fahrenheit, which is 4 degrees Celsius, to 135 degrees Fahrenheit, which is 57 degrees Celsius. As we just mentioned earlier, for those pathogens, the growth is more slowly at a lower temperature, and they have a higher growth rate in the range of 77 degrees Fahrenheit, which is 25 degrees Celsius, to 104 degrees Fahrenheit, which is 40 degrees Celsius. The third aspect to control bacterial pathogens is to kill them using thermal or non-thermal methods. Thermal methods rely on heat, which damage some important components of bacterial cells. The thermal treatment we are quite familiar with is heat pasteurization. If an endospore-forming pathogen is a concern, internal temperature not lower than 240 degrees Fahrenheit, which is 116 degrees Celsius, is needed to destroy the endospores. In order to achieve such a high temperature, pressure higher than atmospheric pressure is needed. Besides temperature, time is also another important parameter for heat treatment. For each heat treatment, each area of the food product needs to be maintained at that particular temperature for a specific period of time to make sure the treatment is effective. Besides thermal methods, some non-thermal methods have also been used in the industry. A commonly used non-thermal processing method is high-pressure processing, which relies on very high pressure to inactivate the microorganisms. Some high-pressure processed food products you can find in the markets include juices, deli meats, and guacamole, and they sometimes have words such as cold-pressed, pressure pasteurized, or pressurized in the labels. Viral pathogens are another big group of pathogens that cause foodborne outbreaks and illnesses. Most of the foodborne illnesses in the United States is caused by viruses. Foodborne viruses can persist in foods and can survive freezing. Different from bacteria, viruses need hosts to grow, and they do not grow in the food products. The most common foodborne virus, which is also the most common causative agent for foodborne illnesses, is norovirus, which accounts for 58% foodborne illnesses in the United States. It is very contagious, and outbreaks usually happen in an enclosed area with a high density of people, such as cruise ship. Common symptoms for norovirus infection include vomiting and diarrhea. Another foodborne virus is rotavirus, which is the most common cause of severe gastroenteritis in infants and young children. There are some other foodborne viruses that have been associated with outbreaks and illnesses, such as hepatitis A and hepatitis E viruses. Proper practices are important in preventing the transmission of foodborne viruses because fecal-oral is the main route for the transmission of foodborne viruses. Patients can shed high numbers of viral particles in their feces and vomit. Some examples of proper practices include practice proper hand hygiene, exclusion of ill food handlers, clean and disinfect services and facilities, and proper disposal of human feces. Foodborne viruses can also be inactivated by processing methods, such as heat treatments or high-pressure processing. Even though foodborne outbreaks caused by parasites are not as frequent as bacteria and viruses in the U.S., those outbreaks still occur. Some parasites are microorganisms, and those are parasitic protozoa, and some parasites are not microscopic, such as parasitic worms, which can be associated with raw meats. Parasitic worms will not be covered in this module. Patients infected by parasites can shed parasites in feces, so food and water can be contaminated by fecal materials from the infected host. Parasites can also infect animal tissues. Therefore, parasites can be transmitted through contaminated food or water, undercooked meats, or person-to-person contact. Since fecal-oral is the main route for the transmission of parasites, proper practices are important to prevent parasite transmission. Freezing and freeze-thaw cycles with certain conditions with specific temperature and time can also destroy parasites. Parasites are sensitive to heat, so heat treatment, such as cooking, can effectively destroy parasites. Here are some examples of outbreaks that are caused by biological hazards. The first example is an outbreak of E. coli 0157H7 infection linked with romaine lettuce started from September 2019 and lasted till December 2019. Romaine lettuce from the Salinas, California was contaminated with E. coli 0157H7. A total of 267 people infected with the outbreak strain of the E. coli 0157H7 were reported from 27 states. 85 people were hospitalized. The next example is an outbreak caused by hepatitis A virus. The virus contaminated fresh blackberries sold at Fresh Thyme Farmers Market and Woodman's grocery stores during September 2019. A total of 20 cases were reported from 7 states and 11 people were hospitalized. The last example here is an outbreak of parasite Cyclospora infections linked to bag salad mix produced by Fresh Express. There were 701 confirmed cases from 14 states, and 38 people were hospitalized.